Thank you. Hi, everyone. Thank you very much for having me, and thank you for a great event so far. My name is Fernandez Benevento. I am a senior bioinformatician working at ZS, and today I'm going to talk to you about some of the different pipelines that we have built, how we address certain problems, and some of the learnings that we have collected along the way. But before I do that, let me give you a brief introduction to who we are. And ZS is a global professional services firm with more than 40 years of impact in healthcare and beyond. I am part of a group within ZS called ZS Discovery, and our focus at ZS Discovery is to work closely with the research and early discovery part of pharma and biotech, and to help them advance their R&D capabilities and successes in areas such as target discovery and data and knowledge management. We use our combined scientific and technical expertise to elucidate novel aspects of disease biology and uncover new target and therapeutic opportunities. So we work with clients that range from large and mid-sized pharma, as well as biotechs, and to make it easier for our clients, as well as for ourselves, and to facilitate the wide variety of projects that we work with, and bring in additional expertise, we have formed a range of partnerships with different providers. One of those partners is Sequera, and this partnership is allowing us to more seamlessly implement end-to-end Nextflow solutions for our clients. It does so by leveraging portable modular pipelines, maximizing value in the cloud, and deploying end-to-end process automation, among other things. Now I would like to show you a brief overview of the type of pipelines that we have developed, as well as some of the needs and trends that we see in the industry. Most of the pipelines that we develop, we build them for client projects to address specific needs. And some of them also interact with each other, meaning that, for example, the output from one pipeline can become the input for another pipeline, therefore becoming whole biological analysis ecosystems. And just to mention a few, and a bit of the trends that we see in the industry, we can talk about multimodal variant calling. And genomics workflows, like variant calling, they are very common among bioinformaticians. There's many workflows out there that follow the best practices. However, for large pharma, it is still important to have a pipeline. When analyzing, let's say, biobank data from hundreds of thousands of samples using whole genome sequencing or whole exome sequencing, then the difficult part becomes the scale. And pipelines are needed in the industry for scale, throughput, reproducibility, and regulatory requirements. We can also talk about pipelines to analyze transcriptomics data, like RNA-seq. And again, RNA-seq pipelines are very common. However, bulk tissue analysis are still important, and we also see a lot of non-human models. Bulk expression profiles obtained from RNA sequencing are very useful, for example, to identify genes that shouldn't be modulated as targets. So for a pharma organization working on target discovery, it's really helpful to have a pipeline that can do this automatically. And while bulk is good, single cell is even better in terms of the amount of biological information you can extract from it. The future integrates both single-cell RNA-seq and imaging analysis to do spatial analysis more accurately, capturing the heterogeneity at a finer resolution, and elucidating biological mechanisms more accurately. Finally, I want to mention protein annotation pipelines and how accurately predicting regions of function in amino acid sequences is crucial for a pharma company which is trying to find targets for a particular disease before they invest their resources into experimentally validating them. So having a pipeline that can process multiple peptides at the same time is super helpful. And while the pipelines we develop are different in nature, they belong to different types of bioinformatics analysis. They do have some things in common, things that are part of our development process, and I would like to highlight some of those. First, let's talk about testing, which is a crucial part of development. And when it comes to the type of datasets that we use both publicly available datasets, which are more commonly used by the community, and also private datasets provided by our clients, which are important because this is the type of data that is more likely going to be run through the pipeline. And while research datasets and clinical datasets are processed pretty much in the same way, they do require substantially different levels of auditability and managed access, which leads to guardrails being employed in different ways. Overall, having multiple test data sets of different sizes and different sources, this will all help ensure that the pipeline is robust and that it can handle different types of data. About infrastructure, we develop pipelines to work both in cloud as well as in on-premises HPC servers. And nowadays, everyone seems to be moving to the cloud, if they're not already there. But in the industry, there's still a lot of legacy HPC servers, so both are important. Although everyone may be moving to the cloud, it can get expensive since there's very high demand of the resources lately. And at the same time, they may already have an HPC local server lying around. So we do see some people moving back to cluster as well. Also, purchasing hardware for on-premises usage is a one-time capital expenditure. And although it may have some downsides when you compare to the OpEx cloud pricing model, it does have some benefits. First, it includes environments where the scientists are familiar working with. And second, the data is stored more cheaply than in the cloud, which is especially important when the data lifecycle policies haven't been established. Fortunately, Nextflow is great for this. Since it's pretty universal, it means you can build a pipeline in one environment and that will run pretty much anywhere. However, a few minor tweaks might still be required here and there, especially when it comes to the resources specification. It's important to know the platform where you plan to run your pipeline because there's likely some costs attached to it, so it's likely that you want to spend some time figuring out the optimal resources and set them up accordingly, do that fine-tuning. Finally, this is not always possible. It depends on many things. But when we can, we like to share back to the open source community, either working directly with the great NF Core community or by sharing the main structures or concepts with Sequera so they can use it as a functional reference. The industry benefits from the sharing and reusability across organizational groups, and having a dialogue with the community allows for the refinement of the workflows. It empowers the clinical teams to use the exact same qualified workflows produced by the research teams, promoting harmonization and reducing analytical batch effects. In all the time that we have spent building Nextflow pipelines, we have come across some challenges that we were able to solve, and here we have summarized a few of those learnings that can hopefully help someone else who is developing Nextflow pipelines. First, because most of the work we do it for clients, we mainly work on private environments. And the thing about private environments is that they always have some particularities, whether that is private infrastructures or databases, firewalls. And the lesson here is that you need to be open-minded, because these particularities will push you to the limits to make you come up with solutions which are maybe not the standard ones used by the community, the one you can find on NF Core, but you will have to come up with a solution which is ultimately specifically tailor-made for your private setup. That's why being open-minded and being flexible is key here. One of the many great things about Nextflow is that you can run every tool that is part of your pipeline from Docker images. You don't need to have them installed on your local or on your organization server. You can just put them on a Docker image or use one that was built by someone else and run it from there. And while those images can be stored anywhere as well, one thing to keep in mind is that the public image repositories, which are very common, they do have some pull rate limits, meaning that there's a limited number of image pulls that a single user can do in a particular period of time. And if that limit is exceeded, the user won't be able to pull in more images and a pipeline will fail. So especially important when you're working on a private setup where the work cannot be stopped because of these limits, it's better to rely on private image repositories. Finally, I would like to highlight the importance of having a thorough, systematic testing process and using different data sets, different sizes. This will help you understand the limits of the pipeline and set up the resources efficiently for a proper cost efficiency balance. One very simple example, let's say you are requesting 32 CPUs for a particular process. You run that on the cloud and that runs fine. But when you run the same settings on a cluster, maybe it doesn't have the 32 CPUs or it has them but you don't have access to them. So then that will make the pipeline fail. So one good practice to do is whenever you're ready to deploy, run your pipeline once on every environment. Make sure it runs successfully. It will save you some time and some headaches from having to fix that on prod. And before wrapping up, I would like to thank Sekera for a great partnership, as well as the great NF Core community and our team at ZS Discovery. And if anyone wants to talk about any of the things that we have discussed here or anything else, please come up to our booth. We would love to have a conversation with you. Thank you very much.
Thank you very much for this wonderful talk, very clear. I'll see if anybody asks, but in the meantime, I was curious, you talked a little bit in that last example about setting resources and testing a pipeline, etc. How much have you run into the problem of needing to balance running really fast versus running really cheap? Because sometimes the amount of resource allocations might depend a lot. If you're running on cloud, like you can give more resources, but it's going to cost you a lot. So is that something that comes into consideration?
Absolutely, yes. It can happen. Like you can almost give more resources and the cloud will run better and you're happy, but then you look at the cost of that and you're like, okay, maybe let's slow it down a bit. Mm-hmm. I'd rather have maybe a longer runtime, mm-hmm. but lower cost. So that balance is important. And maybe you can use the cluster for running faster. It depends. Every case is special, and we look at that.
Yeah, that's a very solid recommendation right there. All right, well, thank you very much. If there's any more questions that occur, please post either in Slack or come to the booth to talk to Andres.